Hi everyone, a very warm welcome to each of you. Today I came with another topic, plant development. This is the part of developmental biology of CSIR NET syllabus. So what we have to study here, let's see. We have to study about the embryo sac development and double fertilization. Then embryogenesis, symmetry in plants, seed formation and germination. Then in part D, organization of shoot and root apical meristem, shoot and root development, leaf development and phyllotaxy, transition to flowering floral meristem and floral development in Arabidopsis and Antirrhinum. This Antirrhinum is snapdragon. Okay, so let's begin with embryo sac development and double fertilization. This one. So before I start with the topic embryo sac development and double fertilization, I will give you some introductory idea about this topic. Then we will go a little detailed in it. So let's begin. So we all know that angiospermic plants are the flowering plants. Okay, angiospermic plants are the flowering plants. Okay, like the fertilization happens in animals. The same process, like the quite similar process, is seen in plants also, where the male gametes and the female gamete will fuse and will form zygote. Zygote will form embryo. Embryo will form seed. Seed will give rise to seedling, and the ultimately whole plant will develop. Since we are talking about angiospermic plants, the plant which is formed will show flowering. Clear? Now, this embryo which is formed, this embryo is a polarized embryo. Okay, that means this embryo has a polarized organization. The apical part of the embryo will form the shoot, and the lower part of the embryo will form the root. Clear? Now, we need to also discuss about the life cycle of angiosperm. So, angiosperm life cycle is haplodiplontic. That means the haploid phase of alters with the diploid phase. So what it is we will discuss. So we need to know that flower have four important parts. I have written that because that it will save our time. So four important parts also known as holes. Okay, what are the four holes? Sepal, petal, stamen, pistil. Pistil is also said to be as gynoecium and stamen is also said to be as androecium. This stamen or androecium is the male reproductive part and gynoecium is the female reproductive part. Okay, so they are the reproductive organs. Stamen and pistil are the reproductive organs. So stamen consists of what? Stamen consists of anther and filament. Okay, this anther is a spore producing organ. Anther produces what? Anther produces the spores named microspores. Another reproductive organ which is the female. So anther is the male one and pistil is the female one. Okay, pistil consists of three important parts: stigma, style, ovary. The ovary is the spore producing organs in female which produces megaspores. So stamens has anther or stamen consists of anther which produces microspore, and this microspore are the male reproductive part and ovary produces megaspore which are the female reproductive spores. Clear? Next we need to discuss about the life cycle of angiosperm which is haplodiplontic. Okay, so what is embryogenesis? The embryogenesis, it transforms a single celled zygote into multicellular microscopic embryonic plant. Okay. So this is just the definition of embryogenesis. So we need to discuss about the life cycle of angiosperm. So it consists of two generations, sporophytic generation and gametophytic generation. So gametophytic generation and sporophytic generation. Now, we also need to know about the characteristics of this two generation. This gametophytic generation, gametophytic generation is a short, nutritionally independent, and haploid phase. Okay, and sporophytic generation is a diploid phase. Okay, now there is an in generation in gametophytic and sporophytic phase. Okay, so what we see the life cycle. So at first, gametophytic generation will produce what? They will produce gametes, the male gamete and female gamete. Okay, they'll produce what? They'll produce gametes. This gamete will show the fusion and will form what? Zygote. The male gamete and female gamete will fuse, it will form zygote. The zygote will undergo mitosis and it will form sporophyte. This sporophyte plant, since they are diploid to form the gametophyte again, they need to show meiosis. So zygote have undergone mitosis. Zygote is also 2N. They have undergone mitosis to form sporophytic plant. The sporophytic plant have to undergo meiosis to form the haploid generation again. Okay, so sporophytic plant shows meiosis and they'll form what? They'll form spores. And the spores will again show the mitosis and form gametophyte. Okay. So gametophytes are haploid. Zygametes are also haploid. There is a fusion known as syngamy. The fusion of male and female gamete is known as syngamy, which will form zygote, which is 2N. Sporophytic plant is also diploid, I told you. Now spores are haploid. See here the meiosis process is occurring. The spores are haploid. The spores will again undergo mitosis. It will form gametophyte. Okay, so this life cycle is known as haplodiplontic life cycle. Clear? Now we will begin our discussion with the sporophytic generation first. Sporophytic generation. So here what we see that the spore producing organs produces spores by the process known as sporogenesis. So sporogenesis is a process where the spore producing organs produces spores. So when microspores are produced, it is known as microsporogenesis. And when megaspores are produced, it is known as megasporogenesis. What is sporogenesis? It is the process of formation of spores. Okay, microsporogenesis is the formation of male spores and megasporogenesis is the formation of female spores. Okay, now so we will begin our discussion with microsporogenesis. In microsporogenesis, what we see that microspores are produced. Where are these microspores produced? These microspores are produced in the organ named as anther. Okay, they happens in the organ of the plant or the whole of the plant named as anther.